Hello and welcome to the SKU Digital Learning Classes. Myself Dr. Gaurav Swaroop Nigam, Department of Botany, Shri Krishna University, Chhatarpur, Madhya Pradesh. Today's lecture topic is transcription. What is the transcription and why it is very important topic for the life science syllabus? Transcription included in the syllabus of class BSc, BSc B.Ed, MSc, Life Science Botany, Geology, Biotechnology, Microbiology, and it is a very important topic of molecular biology. So, what is the transcription? Transcription is a ribonucleotide polymerization where a ribonucleotide get incorporated and form the complementary strand of DNA with the help of RNA polymerase. So, we can define transcription as a process through which RNA molecules form from the DNA molecule. We can say that transcription is a process of ribonucleotide polymerization with the help of enzyme RNA polymerase. So, first important thing is where in the cell the process of transcription takes place. Transcription process in the prokaryotes takes place in the cell, cytoplasm cell, while in eukaryotes it takes place inside the nucleus. Why? Because in prokaryotic cell there is no particular boundary of nucleus inside which the transcription can take place, while in case of eukaryotes there is well developed structure of nucleus, and inside the nucleus the process of transcription takes place, and in this process of transcription DNA is present inside the nucleus, whether it may be prokaryotic cell or eukaryotic cell. So that is the transcription site in prokaryotic and eukaryotic cell. Now, what are the basic features of RNA synthesis? If we talk about the requirement of the RNA synthesis, first we require the precursor. Precursors are the 4 ribonucleotides like the ATP, GTP, CTP, UTP, like adenosine triphosphate, guanosine triphosphate, cytosine triphosphate, and uridine triphosphate. So these are the precursors which are required for the polymerization. Now, polymerization always takes place, as we know, as we studied in the earlier classes, that the DNA is a composition of pentose sugar and nitrogenous base and phosphate group, and the 3 combine together to form a nucleotide, as we are explaining it as a ribonucleotide chain. So one nucleotide, 3' hydroxyl group of one nucleotide attached with the 5' hydroxyl group of the other nucleotide with the help of phosphodiester bond, because between the two nucleotide bond together with the help of phosphodiester bond, and this polymerization takes place to form the RNA from the DNA. So if we see the coding sequences, as we know that if our DNA is a double helical structure and DNA has 2 strands, and these 2 strands read in a 5' to 3' direction and 3' to 5' direction, and it has 2 strands, and both the strands are complementary in nature, and that template which are using as a transcript and for the transcription process is called template strand, and the other one strand is called the non-template strand. So when the template strand in DNA, if the nitrogenous base sequence is T, C, G, A, C, T, then in messenger RNA complementary will be T replaced by A always and C replaced by G always, and in place of T in messenger RNA we always know that T is replaced by the uridine or U. If you think about the DNA molecule being transcribed is a double stranded, and how it get transcribed. So here are the two duplex or the two strand of DNA molecule, and here is the 5' to 3' direction, and three other strand direction will be 3' to 5' direction. So these all are the basics of DNA which have earlier studied in the previous lectures. So how the 5' to 3' polymerization takes place, that is called the elongation process or step of the transcription process. Here we can see that there is a pentose sugar and nitrogenous base, and it, with the help of hydrogen bond, it get attached to the nitrogenous base of the other strand. So there is a base pairing rule with the two strand of the DNA, that adenine is always paired with the thymine and guanine is always paired with the cytosine. Now, what is the requirement? As we have told that there is the requirement of nucleotides, ribonucleotides, CTG, DTG, but there is no requirement of primer. Primer, what is the primer? Primer basically is a strand of RNA which is required for the initiation of transcription process. But in case of transcription, there is no requirement of primer. As we can see in the place of DNA replication, we can see there is requirement of primer, but in case of transcription, there is no requirement of primer. So if we see, there is always other requirement that 5' phosphate and 3' hydroxyl group is also present. So here is same things are present. Requirement, if we see the requirement, template double stranded DNA is required, activated precursor like ATP, GTP, UTP, CTP is required, like adenosine triphosphate, guanine triphosphate, uridine triphosphate, cytosine triphosphate. If we see the divalent metals, these are magnesium ion and magnesium ion. And the important enzyme which initiated the process of transcription is called RNA polymerase enzyme. Now, if we see the structure of RNA polymerase, which is isolated from Escherichia coli by Chamberlain in 1962, we can see the structure. There is a two part. One is core enzyme and one is sigma factor. Core enzyme is formed by the subunit, 5 subunit, 2 alpha, beta and beta dash, and sigma factor. When there is only beta, beta dash and 2 alpha, that part is called core enzyme of the RNA polymerase. And when sigma factor get associated with the core enzyme, it form the holo enzyme. And the complete holo enzyme form structure of RNA polymerase. And RNA polymerase is very and crucial important enzyme for the initiation of transcription. So, if we see the subunit of RNA polymerase, we can see the beta dash, beta, 2 alpha and alpha. Function of beta dash is to get bind with the DNA template, and beta is to also help in the binding of RNA polymerase to the DNA template. 2 alpha, it helps in the assembly of core enzyme, core RNA polymerase enzyme to the primer sequence of the DNA, while the sigma is the recognition start site. It is the very important question for the NEET and other competitive examination question, where it has been asked that the which subunit or which factor of the RNA polymerase is required for the initiation of transcription. So it is the sigma factor of RNA polymerase which recognizes the promoter sequence which is present on the promoter of the DNA. So if we talk about the promoter of a DNA, what is the promoter? As we always told that the gene is a double-stranded part or nucleotide part of a DNA which represent a character, and character always represented in the form of a protein. When gene expresses itself in the form of protein, that would call the process of transcription or gene expression. We can also say that Transcription is the first process of gene expression because it is the central dogma of cell that every gene get transcribed and then translated to form particular protein, and that particular protein represent a particular character like hair color, plant growth, plant length, etc. So that is the transcription pressure. And if we see the steps which are required in the process of transcription, there are three basic steps. One is initiation, elongation, and termination. Initiation first. If we see the gene structure, gene is a part of DNA. So DNA has the promoter region and the coding sequence of the gene. If we see here, we can see the promoter. If we focus on the promoter structure, we find the -35 box and -10 box. These are the base pairs of a DNA. TA box or -10 box is the box where adenine and thymine nitrogenous bases are rich in nature, and this Tata box or TA box is also called the Pribnow box in case of prokaryotic cell, and it is also a very important question for the competitive examination, that the Tata box is also termed by Pribnow box. So it is the structure of promoter of a DNA or gene, promoter gene, in which first part is upstream promoter element and Tata box. Then it is the point after the Tata box or -10 box from where the initiation of messenger RNA polymerization or transcription initiation takes place. This is the structure of RNA polymerase, that how RNA polymerase enzyme using the one strand of DNA, that is called the template strand of DNA, and synthesized with the subunits of RNA polymerase, and the messenger RNA transcript is releases by using the process of RNA polymerase transcription. So here we can see that the holoenzyme or the complete RNA polymerase enzyme scans the double stranded of DNA, out of which one template of DNA strand is using, and the one template strand is moving around the promoter region and recognizes the Tata box where the transcription process gets started. And it is the sigma subunit of RNA polymerase which recognizes the Tata box or -10 box for the initiation of the transcription. So we can see that beyond the +1 side there will be downstream, and beyond the +1 minus, that is called the upstream side of the promoter. So we can see that it is the point, start point, from where the transcription process gets started. That is the downstream +1, 2, 3, 4, and -1, -2, -3, -4, -5, -6, that is called the upstream of the promoter region. Like this one, this is also the slide of the promoter region of RNA polymerase, that is the -35 box, -10 box, and +1 box. -10 box is recognized by the Pribnow box or Tata box. Tata box is why it is the Tata box where transcription gets started. That is also a very important question, because that box is rich in thymine and adenine, and we already know that between thymine and adenine there are 2 hydrogen bond, while in case of guanine and cytosine there is 3 nitrogen bond. So hydrogen bond. In case of guanine and cytosine where 3 hydrogen bond, then the pairing is strong, while in case of adenine and thymine there is 2 hydrogen bond and pairing is weak. So it is very easy to break the 2 hydrogen bond in comparison to the 3 hydrogen bond and get the strand as a template. So we can see there same, the RNA polymerase holoenzyme form the transcript and RNA polymerase form the loop structure. Now next, if there are a structure of 2 gene, we can see here that the template of gene 1 is used for the initiation, or here is transcribed and with the help of RNA polymerase, and there is the formation of transcript. If we see the gene, second gene on the same DNA strand, then the other strand will be the template strand and form the messenger RNA transcript. Same, here is addition with the growing chain. If we see the elongation, which is the next process of transcription, we always see that these nitrogenous bases get incorporated and form the complete chain, which is called the polymerization, where a one nucleotide of 3-hydroxyl group or carbon attaches to the 5-carbon hydroxyl group of the other nucleotide with the help of phosphodiester bond and complete the polynucleotidization. Elongation also, we can also see that there is the first 3' carbon of the first nucleotide attaches with the 5th one of the second nucleotide with the help of phosphodiester bond. So with the help of same, there is also a polymerization of RNA, messenger RNA transcript. So if you see the termination process, as we have said that the process initiate, that is number 1 is the initiation, number 2 is the elongation, and number 3 is the termination. So how the process of transcription get terminated? With the help of 2 steps. Number 1 is Henle loop formation. Through Henle loop formation, this termination takes place and the transcription process gets stopped. And then number 2 is row dependent transcription termination process. So number 1 is Henle loop formation, and how this Henle loop get formed? When the nitrogenous base sequences get repeated after the UUCG, then these sequences form a Henle loop or loop-like structure, where at the base the G and C composition is rich, and these two make the stop to form the Henle loop and get the transcription process stopped. Same with the help of row dependent termination process, and in this process the row enzyme, the row protein, recognizes the RUT site during the process of transcription, and this site recognizes by row dependent or row protein, and this protein stop the process of ribonucleotide polymerization. So these are the steps where the transcription process get stopped. If we quickly recycle the process of transcription, so in first step, as we see that the DNA as a template, RNA polymerase get incorporated with the DNA, and in second step, DNA passes through the elongation process takes place, and the RNA polymerase get released, and the transcript will get released from the process, and the sigma subunit of the process is released, and the transcription cycle complete takes place. Thank you so much.